yung full screen na PowerPoint?
Yes, Kuya.
Okay, okay. So, ang ano natin ngayon, ang topic natin ngayon is primarily on mycology and virology. Basically yung Mycoviro sa board exam, aabot lang yan ng mga 2 to 3 items. So, imagine yun, undergo kayo ng isang SEM sa, wait lang, may mga papasok pa. So, imagine yun, minsan, mag-undergo kayo ng Mycoviro ng 1 SEM only to find out 2 to 3 items lang pala sa board exam. So, usually, bakte ang 65% bakte, mga 67% bakte, tapos mga 28% na para, tapos 5% na Mycoviro. So ganun lang siya kakonti sa board exam. Ganun lang siya kadalang sa board exam. Nevertheless, points are still points pagdating sa board exam. Every point matter na as long as alam mo siya, as long as na-encounter mo siya before, at pag nasagot mo siya, hindi mo na siya problemahin sa board exam. So still, it is a must for us to know yung coverage natin when it comes to Mycoviro. Well, of course, in terms of review, kung time-wise ang pag-uusapan, mag-focus na lang kayo sa bakte kasi marami doon. Sobrang dami doon. Then, ang next nyo i-prioritize is para, tapos after which, yun na, Mycoviro. So, yun yung order of priority recommended for you to study properly ang microbiology na subject natin. So, yun, 20% ang cumulative percentage niya sa board exam, and it is also considered as a major subject sa board exam. So, failure of that subject would lead you to failure to the board exam as well. So, yun. Still, yun nga, malay natin, Mycoviro ang magsasalba sa atin sa board exam. For example, if you are aiming to get 75% na percentage dyan sa micro, ang magpapa 75% pala sa'yo yung nag-iisang tama mo sa Mycoviro, di ba? So malaking yan. Every point matters nga, sabi nila. So, yun. What I'm going to discuss to you today are usually yung increased chance na lalabas siya sa board exam. And of course, in preparation na rin as a review for your comprehensive exam. Nevertheless, sige, mag-start tayo. So for the introduction, study of eukaryotic fungi ang mycology. Their taxonomy, environmental impact, and genetic and biochemical properties. So when it comes to mycology, ang property nila is that in addition dito sa characteristics, they have true nuclei. So meron silang true nuclei. Which makes them classified as eukaryotes. Yan. So yan yung mga they can either be unicellular or they can either be They can either be unicellular or they can either be multicellular. So it depends. So they are non-motile. So basically, pag magtatanong sila bigla sa compre or board exam, motility of Pseudolesceria boydii. So generally wala silang motility, so ang sasagot niyo doon is non-motile. So yun. It is an important thing to know kasi when it comes to motility, ang dapat lang natin pansinin ng mga organisms dito would be bacteria or parasites. So, yun yung mga may characteristic na motility. So, they also have definite cell walls. So, yun. They lack chlorophyll and absorb nutrients. So, madadagdag lang ako ng additional facts dito. They have two phases. So, may two phases ang fungi natin. The first is yung multicellular na mold phase. So, pag mold phase sila, they are multicellular. So, multicellular. And yeast phase naman, they are unicellular. So we get that notion generally na yeast phase um, yung phase ng fungi na kung saan matatagpuan siyang yung phase na yun sa loob ng katawan. So yeast phase yung tawag doon. Usually ganun siya. Pero there are some fungi that exhibits mold phase kahit na nasa loob na ng katawan, ready to infect tissues. So there are different classifications regarding dun sa phases ng mga fungi. So when it comes to fungi that exhibits mold and yeast at the same time, they are called dimorphic. So wait lang. Dimorphic fungi. So mold and yeast. Mold and yeast phase. So yung naman, yung tawag ba dun? Yung specific characteristic ng dimorphic fungi natin is that they are mold at room temp. So usually at 25 degrees. And yung yeast naman natin, they are yeast at 37 degrees and in tissues. So pag present sila sa tissues natin or pag 37 degrees yung temperature ng environment natin, then doon natin makikita yung yeast phase ng dimorphic fungi. So magkaibang situation yung tissue phase ay yung tissue na tinutukoy dito at yung 37 degrees. So for example, pag kinulture mo yung fungi, yung dimorphic fungi na yan, at a temperature of 37 degrees, mag-exhibit siya ng yeast phase kahit nasa labas siya ng katawan, kahit in vitro. At the same time, pag nag-invade siya ng isang tissue, magiging yeast phase pa rin siya. So yun yung ibig sabihin nitong yeast phase at 37 degrees and at tissue. So saka siya mag-exhibit ng mold phase pag under siya sa room temperature, 25 degrees. So yun. There is another classification. We consider this as monomorphic fungi also. So monomorphic fungi. So monomorphic fungi, one good example is Cepedonium. Cepedonium. So which is mold. Mold phase siya. So ibig sabihin monomorphic, either mold or yeast lang, not both. So mold siya at 25 and 37 degrees. So yan yung isa sa example na exemption na sinasabi ko kanina with regards to notion natin sa yeast phase na kung saan tawag mo doon, pag 37 degrees hematic yeast na ganun. So there are some fungi that exhibits mold phase at 37 degrees. Pero generally speaking, pag sinabing yeast phase, usually 37 degrees yan. So yun yung need natin i-consider dyan. We have another classification, what we refer to as diphasic. So diphasic fungi. Magkaiba siya sa dimorphic. So sa diphasic naman, meron din siyang mold and yeast phase, pero the time that they change into that or transform into that phase depends. So kung sa dimorphic fungi, mold siya at 25 degrees, tapos yeast siya at 37 degrees and sa tissue. Whereas sa diphasic fungi naman, mold siya at 25 and 37 degrees. Tapos yeast phase naman siya sa tissues. So yun yung difference nila. Kumbaga, saka lang siya magiging yeast pag nakapasok na siya sa tissue. So itong uri ng fungi na to, diphasic, it is a type of fungi that is considered as mold phase kahit nasa 37 degrees na siya. So the only difference is that saka lang siya magiging yeast pag nasa loob na siya ng tissues. So yun yung difference niya between dimorphic fungi. So wag malilito dimorphic fungi, mold at 25, yeast at 37 and tissue. Diphasic naman, mold at 25 and 37, yeast siya at tissues. So yun yung difference nila, dimorphic and diphasic fungi. So yun. Another thing that we're going to consider about fungi is the presence of hyphae. So hyphae is branching filaments of molds and mushrooms. So ito yung madalas na observe natin sa sa surroundings natin, sa garden ganun, or pag humid yung yung nature ng environment. So madalas natin mapapansin yung mga ganyang klaseng mushrooms. So yun. Um Mass of hyphae or groups of hyphae are what we considered as mycelium. So yan. Before pala that, we have two types of hyphae. So the demaciaceous, dark or hyaline, colorless. So another is septate or aseptate. So ang one good example for septate ay aseptate muna pala. One good example for aseptate is Zygomycetes. Ito yung ito yung isa sa hindi ko trip sa sa Mycoviro, yung medyo wala siyang masyadong analysis na fino-focus. It's more of direct concept siya. So, kung ano yung nandyan sa PowerPoint, yun lang mismo yung kabisaduhin nyo, or kung ano lang mismo ang tinutukoy sa topic ganun. Yun lang mismo. Parang wala siyang reason behind masyado kung bakit siya ganyan, or yun yung medyo hindi lang kaaya-aya sa Mycoviro. Other than that, medyo goods pa naman tong subject na to. So one good example of aseptate hyphae is Zygomycetes. So other term niya, choanocytic. So ito syempre, sabi dito sa septate with cross walls, sa aseptate wala. So no cross walls. Yan. So septate hyphae, all fungi except Zygomycetes. Yun lang need nyo tandaan dyan. All fungi except Zygomycetes. So pag tinanong, all of the following are considered as septate hyphae except. So pag may nakita kayong choice dun na Zygomycetes, yun, ipiliin nyo yun, Zygomycetes. Pero syempre, hindi ganun kadali ang compre. Ang compre is yun yung tanong, pero ang choices mamaya is yung species under sa Zygomycetes. So dapat need nyo rin kabisaduhin yung Zygomycetes niyan. So, yun. Mycelium, mass of hyphae or groups of hyphae. Then we also have two types, vegetative and aerial or reproductive. So, with regards to with regards dun sa positioning nila sa agar plate. So, pag vegetative daw, they are either submerged or on top of the agar substrate. So, parang ganito, itong picture dito. So, either nasa lang siya, nasa loob lang siya ng agar or nasa surface daw. Yun yung ibig niyang sabihin. Wait lang. Whereas sa aerial or reproductive naman, they extend above agar surface. So ito, kaya sila tinawag na aerial kasi nga hindi lang sa agar yung sakop ng growth nila eh. Umaabot pa hanggang sa sa takip nung petri dish. So they extend above the agar surface and may support reproductive structures. So these structures are what we consider as conidia. So more on that details later on. So pseudohyphae naman, a series of elongated blastoconidia made by some yeast. So yun yung need natin consider with regards sa pseudohyphae. So series of elongated blastoconidia. Papakita ko sa inyo mamaya yung picture ng blastoconidia made by some yeasts. So yun. Wait lang, may mga pumapasok pa. Okay? So let's proceed to the different spores or conidia. Asexual spores are what we refer to also as your imperfect fungi. Yan. So asexual spores, they are also considered as imperfect fungi. So, tawag ba dun? We have 4 types here, blastospores. So ito yung blastoconidia na sinasabi ko kanina. So ito yung picture niya. One yeast cell with budding yeast pa na iba. So yan yung yan yung meron sa blastoconidia. So simple budding lang. Ang tawag dito is simple budding process. So yan. So before pala that, yung conidia, there is two classifications dyan. Before tayong proceed sa kabuuan ng blastospores. Conidia has two classifications din. We have macroconidia, and microconidia. Yata na sabi dito sa PowerPoint. So yan. Macroconidia, large of course. So from the term macro, diba? Large multicellular. So medyo important tong macro and microconidia later on. So for microconidia naman, small unicellular. So yun lang. So small unicellular. Meron ding orientations ng conidia. So wait, maganap lang ng sige, dito na lang. There are also orientations sa conidia. Tawag mo doon, catenate conidia. And echinolate. So, for catenate conidia, this is conidia in chains. So, catenate C4C chains. Chains of conidia. So, parang ang itsura niya is one conidia tapos another one. Hilera lang siya. So, ganyan yung itsura ng catenate conidia. So, echinolate naman is rough and spiny. So, mag-base na lang kayo dun sa balik tayo sa hima ng onti. Mag-base kayo dun sa echinocyte. Di ba ang itsura ng echinocyte is spiny siya? Parang ganyan. Sorry. Sa gets nyo na yung point. So, pag sinabing echinolate conidia, rough and spiny. So, we have catenate conidia and echinolate conidia. So, yan yung need nating tandaan kay conidia. So let's proceed na sa blastospores. Yun, production of blastoconidia. So ito, that bud from a mother cell. So it elongates to form pseudohyphae. So yun yung kay blastospores. So we have here poroconidia, phyaloconidia. There's no need to memorize these ones. Trivial lang to, tong poroconidia, phyaloconidia, and aneloconidia. Basta ang need yung tandaan dito is yung apat. So blastospores, arthro, chlamydo, sporangiospores, yung mga yan. So let's proceed na. We also have yung arthroconidia. So fragment from the hyphal strand at the septation points. So ito yung arthroconidia. So with regards to arthroconidia, we have different fungi that exhibits this kind of structure. So arthroconidia. Kindly remember these ones. For arthroconidia, we have here coccygoides. We also have here geotrichum. Geotrichum. We also have here trichosporon. So yan tatlo. Coccygoides, geotrichum, and trichosporon. So yun yung need nating tandaan diyan. CGT. So arthroconidia CGT. So yun, fragment from the hyphal strand at the septation points. We have also here chlamydoconidia. So may 3 types of chlamydoconidia mamaya i-discuss ko yan. Thick-walled survival conidia produced by rounding up enlargement of the terminal hyphal cells. So, there are 3 types dito sa Chlamydoconidia. We have here yung intercalary. So, pag sinabing intercalary, nasa loob lang siya within hypha. So, itong stick dito, yan yung hyphae. Ito. And yung types of Chlamydoconidia will depend on its location relative to the hyphae. So, itong stick dito is what we refer to as hyphae. So, first is intercalary within hypha. Second is terminal at the end of hypha. And the next is sessile. So, side of hypha. So, yan. Nasa gilid sila. So, sessile side, intercalary, so inside, terminal, end. So, yun. Yun na lang yung palatandaan natin dyan. Sa Chlamydoconidia siya, Chlamydoconidia. So, we have 3 types of positioning sa sa Chlamydoconidia. We have sessile, intercalary, and terminal. Intercalary within hypha, sessile at the sides of hypha, and terminal at the end of hypha. So, yun yung tawag doon, different classifications under Chlamydoconidia. So, Chlamydoconidia, actually tinawag siyang survival conidia kasi this conidia is formed in unfavorable conditions. So, kumbaga ang counterpart nito sa bacte is yung tinatawag nating endospores. So, nabubuo tong Chlamydoconidia na to under unfavorable conditions. Kaya siya tinawag na survival conidia. So, yun. We also have sporangiospores. So, internal cleavage of the contents of sporangium, a sac that is supported by sporangiophore. So, ito yung Zygomycetes dapat yan. So, ito yung ina-exhibit daw ng Zygomycetes. So, we have three fungi under sa Zygomycetes. Dati ko nang sabihin. So, we have here Rhizopus. Not type of not fungi under Zygomycetes. These are classifications of sporangiospores pala. So, yun. We have here rhizoid. So, ganito yung tsura niya. Wait lang. So, tawag mo dun. Ganito yung tsura nung rhizoid. Ay, sorry. Mali. Ganyan. Tapos may mga spores dito. Then, nasa baba niya lang mismo. So, yan yung rhizoid. Opposite of sporangiophore. So, ang tawag dito is rhizoid. So, it is opposite. Itong yung tinutukoy niya, yung positioning nito. This position is opposite lang dun sa sporangiophore. Yun yung ibig niyang sabihin. One example of fungi under this rhizoid is Rhizopus. So, at least hindi na kayo malilito sa name niya. So, ito rhizoid again, yung positioning niyan is opposite sa sporangiophore. So, opposite siya dun sa stem. Ito yung sporangium. Itong stem dito yung sporangiophore. Yan nandito pala. So, yung stem dyan. Then, opposite to that is, yung positioning na what we refer to as rhizoid. So, rhizoid lang yan, as in nag-isa. Then, we have also another one. Wala ng space. Burahin ko na lang ito nasa taas. Nakopya nyo na ba ito nasa taas?
Yes po.
Okay. Sige, burahin ko na. So, we also have another, which is yung absidia. Ang tawag naman dun is absidia. So, ito yung sporangium. Ito yung sporangiophore. And then, 2 rhizoids at both ends. So, yan yung absidia. So, rhizoid yung tawag dito. Tapos, sorry, mali. Rhizoid yung tawag dito sa tatlong something na dito. And yung tawag dito sa positioning niya is rhizopus, not the fungi itself. So, rhizopus yung tawag. Then, ang tawag dito, two rhizoids at the opposite ends are absidia. So, yun. Rhizopus yung yung position ng rhizoid opposite to sporangiophore. Whereas absidia naman is alternate rhizoids and sporangiophore. So, rhizoid, sporangiophore, and rhizoid. So, alternate, absidia. Then we also have here another is mucor. So, mucor. Wala siyang rhizoid. Yun yung need nating tandaan dyan. And all of these are unique to Zygomycetes. So, sporangiospores. Again, ulitin ko lang for memory's sake. Rhizoid, yung tawag dito sa position, dito sa parang tatlong linya dito. Rhizopus yung tawag dun sa positioning ng rhizoid just opposite to sporangiophore. Absidia naman is yung rhizoid na alternate dun sa sporangiophore. So at both ends. So rhizoid, sporangiophore, and rhizoid. Mucor naman is yung wala siyang rhizoid. So yun yung sporangiospores. And it is unique again to Zygomycetes. So proceed tayo sa sexual spores or perfect fungi. Uh, Spores or conidia under sexual spores. So we have here zygospores, ascospores, basidiospores, and oospores. So for zygospores naman, it is fusion of two compatible hyphal arms and are surrounded by a thick-walled protective zygosporangium. So ito, from the term zygospores, so it is also exhibited by Zygomycetes or Zygomycota. Wala siya dun sa wala siya dito sa PowerPoint yung pictures, pero na-search ko naman siya, ganito yung tsura. So, para siyang candy wrapper. Ganyan. So, ito yung mga spores sa loob. So, fusion of two compatible hyphal arms. Ito yung hyphal arms, tong dalawa. So, hyphal arms. And are surrounded by a thick-walled protective zygosporangium. So, ito yung zygosporangium, yung thick wall. Ito yung hyphal arms, tong dalawa. Ito yung zygosporangium, itong wall dito. And ito yung mga spores. So, yan. Zygosporangium to, itong wall, itong dalawa dito hyphal arms and spores yun nandito sa loob. We also have ascospores. So, 8 ascospores are inside an ascus. So, ganito lang yung tsura niya. Ito yung ascus. And at least may 8 daw na ascospores. Yan. So, and maybe surrounded by a protective ascoscarp. So, there's another protective layer we refer as ascoscarp. And ascomycota yung tawag sa mga fungi that exhibits this kind of spore. We also have basidiospores. Ang itsura niya is this one. So, they protrude out of a mother cell called a basidium. So, ganito ang itsura niya. Para siyang yan. So, ito yung basidiospore. Okay, hindi na kasha. So, basidiospore yan. And itong mother cell is basidium. So, yan yung yan yung tsura niya. Ang description pala sa ascospore is sac-like. So, sac-like ascus. Yun. Basidiospores naman club shape. Nabanggit ba dito? Hindi siya nabanggit. Pagpasensya nyo na, di kasi masyadong explanatory yung mga um, terms regarding dito sa mycology. So medyo direct yung approach niya. Kaya medyo binabasa ko lang din yung ibang terms. Pero for you to get the picture din, yun, ini-illustrate ko na lang din yung mga itsura nila. We also have oospores, involve the fusion of cells from two separate non-identical hyphae. So, fusion of cells from separate non-identical hyphae. So, ganito yung itsura niya. So, kung yan yung basidiospore, ganito yung itsura ng oospore. Wait lang. So, ganito yung itsura ng oospore. Tapos, malalaki yung bola sa loob. Yan. So, yan yung itsura ng oospore. Oogonium yung tawag dito sa wall. Tapos, tawag dito sa loob yung mga spores dyan, yung oospores. And, itong tawag dito sa stem that supports the oogonium is yung tinatawag nating anteridial hypha. So, yan. So, if hindi natin makabisado yung definition nila, at least yung drawing na lang mismo. So, pag sinabing hyphal arms, isipin nyo candy wrapper siya, ito yung tsura. Pag sinabing sack-like ascus, parang nasa loob lang ng sako, wala yung tali, ganun. Club shape naman to, pero mukhang trident shape. Pero anyway, yun naman yung sabi sa books. So, club shape. Tapos ganito yung tsura ng oospores. So, medyo similar sila sa ascospores, pero ang difference is mas malalaking bola sa oospores. And meron siyang stem. So, yun yung difference. So far, nakaka-follow ba?
Yes, sir.
Sige, pasensya nyo na unholy hour ngayon eh. So, yun. Okay. Media and tests. So, ang isa sa pinaka madalas na ginagamit na culture media pagdating sa sa fungi, sa mycology is Saborodextrose agar or SDA. So, it inhibits the growth of many bacteria. Quick recall lang. Ang normal pH na kung saan nagre-reside ang mga most pathogenic bacteria is between pH 5.5 to 8.0. So usually ranging from 6 to 8 na rin. Kaya hindi ideal sa kanilang tumubo dito sa Saborodextrose is because yung pH nila konti lang, yung parang nasa minimal lang siya, range lang siya between dito sa 5.5 to 8.0. And thus few bacteria only can grow within this agar. So another is yung pH is favorable sa most of fungi. So the problem here is sa SDA hindi tumutubo si Histoplasma capsulatum and some strains of Nocardia asteroides. So Nocardia asteroides, if na-encounter na natin siya before, it is a type of actinomycetes. Pag sinabing actinomycetes, these are fungi-like bacteria. Actinomycetes. Yan. So yan yung yan yung classification nila Nocardia. La Streptomyces yung mga yan. Actinomycetes yung mga yan. So usually Saborodextrose agar is also considered as non-selective kasi majority ng fungi parang siya yung nutrient agar equivalent ng bacteria. So SDA is yung nutrient agar equivalent niya. Yun. Wait lang for a while. Okay. And we also take note na majority sa fungi, mycology, yung culture natin, it's not it's not really ideal para sa atin na patubuin sila sa 37 degrees. Ang recommended by Baylis is 30 degrees. So, usually ito yung highest daw. high range ng room temp. Still under room temp, pero ito yung pinainit na room temp, 30 degrees Celsius. If yan yung yan yung estimated na temperature required for fungi to grow. Pero pag range ang tinatanong, uh, 25 to 30 degrees. So yan yung range. Culture can be held also for within 30 days. So culture can be held within 30 days. Pero ang sabi ni Baylis, 21 days. So according to Baylis, since si Baylis na yung basis ng board exam, 21 days. Yan. So if kung ako lang yung kung sa akin lang ang susundin ko is yung 21 days. Okay? So next, proceed tayo sa SDA with cycloheximide and chloramphenicol. So this agar is suitable for those specimens involving skin, hair, and nails. So basically ang population ng fungi involved dito are your superficial mycoses. So yung skin, hair, and nails na mga fungi natin. Superficial mycoses. So yan. There are some fungi that are being inhibited by cycloheximide. Sige, kalimutan nyo na lahat na nandito. Wag lang si trichosporon and si Cryptococcus neoformans. So yan, kahit silang dalawa lang, wag nyo kalimutan. Cryptococcus neoformans, they are inhibited by cycloheximide and trichosporon as well. So ang purpose ni chloramphenicol dito is that it inhibits the growth of bacteria dun sa culture media na yan. So suborodextrose agar with cycloheximide and chloramphenicol. So basically, yung SDA dito, this is now considered as your selective media. So selective lang siya. Selective inhibitory media. Kasi it inhibits the growth of certain populations and at the same time, it allows the growth of some specific populations as well. So yun yung need nating tandaan. Basta ang key term lang natin sa culture and tawag mo dun? Sa culture media ng requirements ng mga microorganisms is that pag nilagyan mo siya ng antibiotic or any form of of inhibitory substances, then doon siya magiging selective. So pagkitandaan 'yun, doon siya magiging selective. Pag naman, pag nilagyan mo siya ng mga special nutrient requirements, doon siya magiging enrichment. So enrichment media. Or pwede ring enriched media. May difference yun eh. Medyo nakalimutan ko nga lang. Pero may difference ang enriched media sa enrichment. So yun. Pag sinabi namang pag naglagay ka ng pH indicator doon, and at the same time, sugar, mga ganyan, some substances that require differentiation between different microorganisms, then that's what we refer to as differential culture media. So, yun yung mga key terms na need nating tandaan. So, the mere fact na itong SDA na tawag doon, non-selective by nature, nilagyan natin ng cycloheximide and chloramphenicol, that makes it selective to some fungi only. So, yun. BHI, so brain heart infusion agar, it supports the growth of bacteria and fungi including histoplasma and tocardia. So dito naman babawi yung yung dalawang hindi pwedeng tumubo sa SDA. So for example, may case study. A medical technologist inoculated for fungal studies yung BHI ay yung suborodextrose agar. Naglagay siya ng nag-inoculate siya from the specimen doon sa suborodextrose agar. A few days later, wala pa rin growth doon sa SDA. So what is one of the main reasons for this? So pag ang sinabi doon, if the suspected microorganism is Histoplasma capsulatum. So yun yung correct answer. Kasi you cannot grow Histoplasma capsulatum sa suborodextrose agar. But you can allow its growth sa BHI agar. So with blood, yan, histoplasma and nocardia. Recommended for converting dimorphic fungi. So this culture media is important if we are going to appreciate the transformation of one phase to another pagdating sa dimorphic fungi natin. So from mold to yeast, yung mga yan. From yeast to mold, whatever. So basta yun, conversion of dimorphic fungi. Reinhardt infusion agar with blood. So ito, BHIA with blood, cycloheximide ulit, and chloramphenicol. Again, ang purpose ng chloramphenicol is it inhibits the growth of bacteria. Cycloheximide inhibits the growth of some fungi. So yun. Para mas maging selective pa siya. So this one, it also inhibits the growth of nocardia. Kanina it allows yung pag solo BHI agar lang, ina-allow niya yung growth ng nocardia. Pero lagyan mo siya ng selective substances, may inhibit yung growth niya. So this is also ideal for dermatophytes or yung superficial mycoses natin. Isolated from skin, hair, and nails. So ngayon, based from this slide, yung knowledge natin from this slide, yung mga nabanggit na culture media dyan, pag nilagyan mo sila ng cycloheximide, they are usually for dermatophytes. Okay? Ulitin ko ha. If nilagyan sila ng cycloheximide and chloramphenicol, they are usually for dermatophytes. So, yun. Ano pa ba? Sige, magbabanggit pa ako ng ibang culture media later. Do you follow?
Yes, sir.
Kaya pa? Konting slides pa lang to. Kaya pa ba? Hello? Okay. Sige. So, proceed na tayo sa next. Cornmeal twin 80 agar. So, demonstrates blastoconidia. So, yan. Blastoconidia, ano na ulit sila? They are asexual spores, di ba? They bud off from a mother cell. So, yun yung characteristics ng blastoconidia. So, demonstration of blastoconidia, pseudo-hyphae, arthroconidia. So, yung arthroconidia, balikan natin. Diba ang arthroconidia is fragment from hyphal strand. So, I forgot pala to emphasize kanina yung tsura nila. So, ganito yung arthroconidia. Parang may mga spaces in between. So, yan yung meron sa arthroconidia. Ang tawag pala dito sa spaces in between, ito, kalimutan ko sabihin, disjunctor cell. So, wait. Tama. Disjunctor cell. Yan. Disjunctor cell. So, yun. And chlamydospores. So, yan. In the identification of Candida. So, usually ang cornmeal twin 80 ginagamit siya for Candida. Potato dextrose agar naman, enhanced sporulation and pigmentation. So, ang need nating tandaan kay potato dextrose is it is preferred as a subculture rather than a primary. So, ano bang difference ng primary plating sa subculture? Primary plating medium is yung culture media na ginagamit natin pagkagaling mismo sa specimen yung i-inoculate natin. Pero pag sinabing subculture medium, sila yung culture media na nagaling mismo yung inoculate sa primary plating medium. Yun yung difference. So for potato dextrose agar daw, mas prefer siya as a subculture medium kaysa sa primary plating medium. So yun. We also have inhibitory mold agar. Hindi naman masyado important yan. Mycocell agar. So isa lang yung pinapatandaan ni Sir Raymel dito. Dermatophytes. So yun lang. Yun lang need nyo tandaan dyan. We also have here dermatophyte test medium. So isolation of dermatophytes. Ganun lang din. Okay, so for birdseed agar, wag niyong kakalimutan. Pag sinabing birdseed agar, Cryptococcus neoformans agad ang unang papasok sa isip niyo. So, ang mnemonics ko diyan, ang creepy ng ibon, kaya siya tumae ng seed. So, yun yung yung definition ko diyan. Ang creepy ng ibon, kaya siya tumae ng seed. So, birdseed agar for Cryptococcus neoformans. Um, sa reference ni Sir Rimmel, may other term siya, caffeic acid agar. Pero sapat nang malaman niya birdseed. So, birdseed agar is a differential enrichment medium. So, enrichment medium. So, differential, pag sinabing differential, meron siyang specific component na nagpapa-identify sa Cryptococcus neoformans, sorry. And enrichment naman is para meron kang nilagay dyan na substances that support the growth of Cryptococcus neoformans. So, birdseed agar is therefore differential enrichment medium. So, we also have trichophyton test agars, speciation for trichophyton species. So, from the name itself. And germ tube. So, wag natin kakalimutan tong germ tube for Candida albicans. So, wait lang ha. May specific notes at ako dyan. For a while. Ayun. So, ito yung general na steps for us to determine yung um, mga protocol na required para sa pag-identify ng candida. So, or hindi lang sa candida, there are some fungi also that also requires these steps. For first step is germ tube. So, for germ tube, this involves yung yung identification ng Candida albicans. So, magpa-positive si Candida albicans and and dubliniensis. Candida dubliniensis. So, positive sa germ tube si Candida albicans and Candida dubliniensis. Second step, to differentiate those two, morphology on cornmeal agar. So, morphology on cornmeal agar. So, this differentiates yung yung Candida albicans from Candida dubliniensis. So, for Candida albicans, enhanced chlamydoconidia siya. So, for Candida albicans, enhanced chlamydoconidia. So, yan. Enhanced chlamydoconidia. So, for third step naman to further differentiate other fungi from other fungi din, we also have carbohydrate utilization. So first, hindi lang kay Candida tong mga to Ito yung general steps sa sa fungal testing. So carbohydrate utilization for a while. So carbohydrate utilization. So for the first step, we have germ tube. Second step, morphology on cornmeal agar. And third step is carbohydrate utilization. And this carbohydrate utilization is the most common conventional definitive identification. Conventional definitive identification. So yan. Ulitin ko lang si carbohydrate utilization, it is the most common conventional definitive identification. So first step, ulitin natin, germ tube. Second step, morphology on cornmeal agar. Third step, carbohydrate utilization. So yun. Most common conventional definitive test for Candida, for fungi. Okay. So sa urea agar slant, it demonstrates urease production, obviously, kasi urea agar nga. May mnemonics dito na tinatawag nating T-punch. So this does not involve mycology. This also involves bacte. So T-punch. Yan. For T-punch, T stands for Trichosporon mentagrophytes. T mentagrophytes. Trichophyton pala, sorry. Um, Trichophyton mentagrophytes. We also have PPM. Kung maalala nyo sa Enterobacter. So, Enterobacteriaceae, PPM stands for Proteus, Providentia, and Morganella. We also have urea plasma. So, miscellaneous bacteria siya. From the term urea plasma, so positive siya sa urease, sa urea agar slant. So, ito yung mga positive sa urease, guys. We also have nocardia, so actinomycetes. To differentiate nocardia from the rest, gawin lang natin saborodextrose agar yung culture natin. We also have cryptococcus. So to demonstrate kung cryptococcus yung nanap nating species, i-culture lang natin sa birdseed agar. Then we also have Helicobacter. So this is for Helicobacter naman, ito yung isa sa best na identification or differential test between Helicobacter and Campylobacter. So yun yung isa sa main na pang-differentiate between Helicobacter and Campylobacter species, urease. So positive si Helicobacter, negative si Campylobacter. Okay? So T-punch, remember that mnemonics. T mentagrophytes, Proteus providentia morganella, urea plasma, nocardia, cryptococcus, and Helicobacter. So, yun. Wag nyo lang isipin itong mga nandito. Mag-focus na lang kayo sa T-punch. Clear ba?
Sir, urease po ba or urea agar slant yung nagdi-differentiate ng Helicobacter sa
Urease,
Campylobacter?
urease. Urease.
Sir,
Okay. Pero given kasi na yun nga, yung urea agar kasi pwede siyang plate, pwede siyang slant or tube. So, mas magandang itong T-punch is for urease. Basta as long as ilagay mo siya sa isang urea agar, magpa-positive 'yan. Yun yung ibig kong sabihin. Pero yun nga, Helicobacter urease production yung best na pang-differentiate niya with Campylobacter. Okay? So next, we have here hair perforation or baiting test. Ito useful siya for tawag mo dun sa mga dermatophytes natin. Okay. So, T. rubrum versus T. mentagrophytes. Positive si mentagrophytes sa hair baiting, negative si T. rubrum. So, what's another differentiation between T. mentagrophytes and T. rubrum? Siyempre yung urease, di ba? Kasi nabanggit natin kanina, T-punch, ang nandun lang sa T is T. mentagrophytes. So, negative si T. rubrum din dun. Yan, nandito na pala. So, it is also used as a point of differentiation between Microsporum canis and Microsporum equinum. So, yung hair perforation. Positive si canis, negative si equinum. So, equinum from the term equi, kabayo. So, isipin nyo na lang dyan na kalbong kabayo, kaya siya negative sa hair perforation. Okay? So, yun. Urease test. T. mentagrophytes positive versus T. rubrum negative. So, Cryptococcus is also positive. Yun, urease test, yun, ulitin lang natin, T-punch. Yan. Thiamine requirement, not that much important. Growth on rice grains. So, ito yung pang-differentiate between Microsporum canis versus Microsporum odwini. So, yan. I-ano natin siya mamaya. I-differentiate pa natin siya further mamaya. So, positive si M. canis, it turns the rice grains brown. Yan. Germ tube test, presumptive identification of Candida albicans or Pseudobliniensis. So, kanina balik tayo. Sinabing presumptive. So, dito ginamit si albicans versus si Dubliniensis. Positive silang dalawa dyan. We also have levodopa, ferric citrate test for Cryptococcus and carbohydrate utilization. Okay. So, microscopy naman tayo ngayon. Ito natanong to sa sa board exam. Recalls ko noon sa January. Which of the following is the most commonly used microscopic method for fungal elements? Ang choices are saline wet mounts, lactophenol cotton blue, potassium hydroxide, and gram stain. The correct answer here is potassium hydroxide. Yan yung ginagamit for hindi lang fungal elements for uh, dermatophytes yung yung tanong dun. So ito talaga yung ginagamit, potassium hydroxide. Kasi yun nga, it dissolves the keratin. Di ba hair, skin and nails, they are filled with keratin. And this can impede your vision or your field of view pag mag-review na tayo ng specimen involving fungi. So we need potassium hydroxide to dissolve keratin. Kaya siya yung most commonly used na microscopic method or slide preparation method with regards to dermatophytes. So 10 to 20% potassium hydroxide. Well, actually kahit potassium hydroxide lang sapat na, kahit wag na yung percentage. Pero usually ang ginagamit na percentage is 10. So yun nga, kahit potassium hydroxide sapat na. So wag niyong kakalimutan yan, it dissolves the keratin. Kasi kung saline wet mount ang gagamitin mo, saline wet mount, walang kwenta yan with regards to differentiating fungal elements kasi nga malay natin yung isang buong kuko na yan, tinakpan niya yung fungi na dapat mong ma-visualize. So yun, dapat mo i-dissolve yun by potassium hydroxide. Okay, so yan. Saline wet mounts for budding yeast, hyphal elements, particularly for vaginal specimens. So yan na lang yung tatandaan nyo for saline wet mount. So pag sinabing vaginal specimens, so pwede tayong maka-observe dito ng fungi involving dun sa genito-urinary tract ng mga babae, which is yung candida. So yun. Also observable on candidiasis. And particularly present yung disease na yan sa mga patients who are immunocompromised. Particularly those patients with AIDS. So yun, candidiasis. And ang isa daw sa madalas na preparation na ginagamit for vaginal specimens would be saline wet mounts. We also have lactophenol cotton blue. So it stains fungal isolates. Yan. May purposes yung phenol, yung lactic acid and cotton blue dyan. Yan. Sorry, hindi ko nakikita yung pointer ko. Yan. Phenol kills organisms. So, yung phenol dyan. Lactic acids preserves fungal structures. So, pinapatay ng phenol lahat ng organisms na present dyan sa slide. Kaya lang, para ma-preserve pa yung cellular detail, doon papasok si lactic acid. And cotton blue stains the chitin in the fungal cell walls. So, yun. We also have gram stain. So, usually they are gram positive daw. Pero yung gram stain madalas ginagamit sa bacteria yan. So, no need to dwell much on that. Acid fast stain. So, there are some fungi that exhibits partially acid fast stain. Actually, there are some authors na nagde-debate kung fungi ba talaga Synocardia or bacteria. Pero ang mas proper term sa kanya is Actinomycetes. So, fungi-like bacteria. Kaya lang, ito ang Synocardia, kaya siya nalagay dito kasi yun nga, it exhibits fungi-like characteristics. And at the same time, positive siya sa um, partially acid fast siya. So yan. Ang mga partially acid fast na non-mycobacterial food na bacteria. So ang mnemonics ko dyan is it's nice and good to be RMT. So yan, for a while. Okay. Sorry, nagbigay kasi sila ng announcement sa med. Anyway, yun. So for itong mnemonics na to, it's nice and good to be RMT. Yan yung mga partially acid fast na mga bacteria. Partially acid fast meaning it's not as acid fast as mycobacteria, pero it demonstrates acid fast property. So yun. Nice dito means nocardia. So nocardia. Good dito gordonia. R dito rhodococcus. M dito, well actually ito yung mga partially acid fast or acid fast na. Actually nilagay ko lang si mycobacteria dyan para lang masabing acid fast din. Mycobacteria. Pero if talagang isipin mo kung sino-sino yung mga acid fast, it's nice, good, R, and T. So, it's nice and good to be RT ka, Fen. Hindi it's nice and good to be RMT kasi medtex kayo eh. So, nilagay ko siya dyan. And, Chucamurella. So, yan. It's nice and good to be RMT. They are your acid-fast organisms. M there stands for mycobacteria and it is truly acid-fast, whereas yung nice, good, R and T, they are your partially acid-fast. So, yun. India Inc., particularly isang organism lang need natin tandaan when it comes to India Inc. See? Cryptococcus neoformans. So, India Inc. is a negative staining. Pag sinabing negative staining, yung background yung ini-stain, hindi yung organism. So, yun yung negative staining. We also have periodic acid shift because basically, fungi or fungal cell walls and their cell membranes contain a lot of sugar molecules. Periodic acid shift kasi they are specialized to stain carbohydrates. Kaya expect na magpa-positive ang fungi dyan. So, ang positive result is magenta. Pero not as much as important sa mycology. Tandaan nyo itong pass sa histopath, wag sa myco. Calcofluor white stain naman is sensitive stain. Yun lang nabanggit dyan. Wait lang. Check ko if meron akong notes dyan. For a while. Wala. Ang sinabi lang dito sa calcofluor white stain is that ginagamit siya sa fluorescence microscopy. Yun lang yung side notes ko dyan. Fluorescence microscopy. So, yun. Then we also have Hacker modification. So, parang Hacker modification of gram staining lang din yan. All fungi are gram positive. Okay, so before proceeding to superficial mycoses, are there any questions sa previous topics natin?
Wala po, sir.
Okay, sige. Sige, superficial mycosis na tayo. So, wag kayong malilito Itong mga nandito is yung mga sakit. Ito mga nandito is yung mga etiologic agents. So, yung mga causative agents. Ito yung isa sa hindi ko gusto nung review. Yung mga pangalan sa mycology. We have Theriasis versicolor, Malassezia fulfur. Yan. Tinea nigra, Hortea wernicki. So parang nagbabanggit ka lang na Harry Potter spells na abnormal. So yun. White Piedra, Trichosporon species, Black Piedra, Pedrea hortei. So yun yung mga need nating tandaan dyan. One characteristic, of superficial mycoses is that they do not induce cellular response, cellular immunity. So yan yung advantage ng mga fungi under this one. Walang kwenta ang cellular immunity ng host kasi wala silang masyadong access sa ini-infect nitong mga to. Basically because their area of pathogenicity involves only the superficial areas of our body such as skin, nails, hair. Sa tingin ba natin merong mga T-cells na pupunta dun sa skin, hairs, and nails niyan? Wala. So basically, walang cellular immunity dito sa superficial mycosis natin. So, Theriasis versicolor, definition na nandito sa side notes ko is it causes uneven pigmentation. Of skin. Yan. So, causative agent, Malassezia fulfur. So, Malassezia fulfur, isa sa need nating tandaan sa kanya is it loves oil. So, particularly olive oil. Olive oil loving fungi. So, yan. Malassezia fulfur. So, for those individuals who are suffering from Malassezia fulfur at nagkataon na mahilig sila magluto or what, at nagkataon na madalas silang gumagamit ng olive oil, they can also involve other individuals sa infection. Kasi Malassezia fulfur loves olive oil daw. So, yan. For Black Piedra naman, brown or black crust. So, sulat ko na lang. Hindi ko alam kung meron siya dito sa PowerPoint. Hindi ko pa masyado nakikita tong PowerPoint. Brown and black crust outside hair shaft. So, yan. Piedrea hortei or Hortea, Piedrea hortei. Yan. White Piedra naman, light brown nodules on beard. Light brown nodules on beard. So, si Trichosporon species ang involved dyan. Okay, so yan lang yung need natin tandaan for superficial mycosis. So, white piedra, light brown nodules on beard caused by Trichosporon. Black piedra, brown and black crust outside hair shaft. And Theriasis versicolor, uneven pigmentation of skin. Unfortunately, wala akong side notes sa Tinea nigra. So, yan. Cutaneous mycosis naman, these are caused by fungi that infect only the keratinized tissue. So, skin, hair, and nails. Trichophyton, microsporum, and epidermophyton. So, cutaneous mycosis. Superficial mycosis naman to nandito. We have anthropophilic, epidermophyton, flocosum, yung mga yan. So, basically, more on memorization yan. Pero ang need natin tandaan dito is yung sa may antro, syempre. Kasi pag sinabing antro, man, and yun yung focus natin mga medtech. So, epidermophyton flocosum, trichophyton mentagrophytes, rubrum, tonsurans, yung mga yan. Need din natin tandaan itong gypsum. Geophilic. And itong zoophilic naman is Microsporum canis. Yan. So we also have subcutaneous mycoses, traumatic inoculation to skin or subcutaneous tissues. So basic recap lang sa histology natin. Three basic layers of skin: epidermis, dermis, and subcutaneous layer. So imagine nyo yung lalim yung ini-infect ng mga fungi. So lesions become granulomatous and expand slowly from the area of implantation. So we have here sporotrichosis, Itong sporotrichosis is yung tinatawag nating rose gardener's disease. Yan. Mycetoma, Chromoblastomycosis, and Hyphae. So, wait lang. Ay, hindi niya diniscuss yung mga nandito sa ano. Sige. Nagdagdag na lang side notes dito sa mga superficial whatnot. So, superficial mycosis, by now, pinapamemorize ata sa inyo yung different types of tinea or ringworm. So, we have Tinea capitis. It involves scalp. Capitis, scalp. Tinea barbae. It involves beard. So, Tinea capitis, scalp. Tinea barbae, beard. We also have Tinea corporis. So, corporis, ibig sabihin body. We also have Tinea cruris, which means or jack itch, so groin. So Tinea corporis body or Tinea cruris groin. So other term niya daw is jack itch. So ito yung mga different ringworm infections. We also have Tinea unguyum or nails. It infects the nails. We also have Tinea pedis, so foot. Yan. Ito yung tinatawag nating athlete's foot. So yan yung mga different ringworm infections. Ulitin lang natin, Tinea capitis, scalp, barbae, beard, corporis, body, cruris, groin, unguyum, nails, pedis, foot. So yun. Tinea cruris is also known as jack itch and Tinea pedis is also known as athlete's foot. So sa naman, sa cutaneous mycosis naman, we have here yung tawag ba dun? Trichophyton, Microsporum, Epidermophyton. Ito yung need talaga nating tandaan with regards to dito sa mga to. Pag sinabing Microsporum, skin and hair. Lumalabas sa board exam. Which of the following microorganisms infects only the skin and hair? Which of the following organisms infects the skin and nails? So, ito yun. Epidermophyton. And ito pa. Ilang beses na siyang lumabas sa board exam. Wag niyong kakalimutan to. Epidermophyton, wala siyang which of the following superficial mycoses does not exhibit microconidia? Epidermophyton yung sagot. So yan. Pagkitandaan, microconidia. So wala siyang microconidia yung epidermophyton. Club-shaped macroconidia without the presence of microconidia, si epidermophyton. We also have ito, trichophyton, from the term tri. So mag-base na kayo dyan. Skin, hair, nails. So yan. Yan yung ini-infect niya. Trichophyton. So skin, hair, nails. Microsporum, skin and hair. Epidermophyton, skin and nails. It also exhibits no it exhibits no microconidia or does not exhibit microconidia. Yung trichophyton naman, tatlo, skin, hair, and nails. So yan yung mga need nating tandaan dyan. Okay. Now, for differential tests involved dito sa cutaneous mycosis natin, nabanggit na natin yung sa T. rubrum and T. mentagrophytes. So, yan. Ang pH indicator ng dermatophyte medium is phenol red. So, phenol red yung ano, yung... Ba't yung mga ngayon lang sumasabi? Okay. So, phenol red yung ano, phenol red yung sa dermatophyte test na culture media. So, sino-sino ulit sa, balikan natin sa micro, sino-sino ulit yung mga ano, mga may phenol red na culture media. So, actually ano ang mnemonics ng review center namin dito is MX-cut, pero ang mnemonics nating mga CSU1s dito is Texmucidin. Yan. So, Texmucidin. T stands for TSI. X stands for psilocybin desoxycholate. M stands for mannitol salt agar. U stands for urease agar. Or similar lang siya dun sa ano sa urea agar slant kanina. C stands for wait lang. I-chat ko lang 'to mamaya. Medyo hazy na siya sa utak ko. D stands for dermatophyte. So, yan yung mga need nating tandaan dyan. So, yun. Yan yung sa dermatophyte test mincha. And for T. rubrum versus T. mentagrophytes, na banggit natin kanina, with regards to hair baiting test and sa urease, ang nagpa-positive dito is si T. mentagrophytes. Ang nagne-negative dito si rubrum. Yan. We also have T. tonsurans and T. schwenleini. So, ang isa natin tandaan kay schwenleini is fabric chandelier. So, antler-like hyphae or fabric chandelier. Wait lang. So, si T. schwenleini. Fabric chandelier. And ang fabric chandelier is meron daw siyang description na antler-like hyphae. So yan. T. schwenleini or exhibits fabric chandelier. Huwag nyo kakalimutan yung fabric chandelier. Lumalabas yan sa compre. Wala na kasi sila masyadong matanong sa Mycoviro. Kaya usually yan yung tinatanong nila. Fabric chandelier schwenleini. Tonsurans naman, balloon forms. Balloon formed microconidia. So yan. Balloon formed microconidia. So sino na ulit yung yung fungi na yung cutaneous mycoses that does not exhibit microconidia? Si epidermophyton. Huwag niyo kakalimutan yan. Sino yung bacteria daw? Sino yung cutaneous mycoses na nag-exhibit ng fabric chandelier? Si Schwenleini, Trichophyton schwenleini. So, how will you differentiate mentagrophytes from rubrum? Hair baiting test and urease test. Positive si mentagrophyte, negative si T. rubrum on both tests. So, yun. Sa hair baiting test pala, nakalimutan kong sabihin kanina, ang positive result for hair baiting test is V-shaped penetration of hair shaft. Shucks. So yan. V-shaped penetration of hair shaft. Okay. Wait lang. May nakalimutan din ako sabihin kay Malassezia furfur. For Malassezia furfur, sa microscopic examination niya, spaghetti and meatballs appearance. So, huwag natin kakalimutan yan. So, for example, kung mahilig kayo magluto, ganun. Tapos may na-discover kayo na special modification ng spaghetti and meatball. Tapos kayo yung unang nakadiscover doon. Pwede niyo siyang pangalan ng Malassezia, mga ganyan. So, mukhang pinasosyal, pero yung pala, pangalan na pala ng fungi yun. So, yun. Okay, so proceed na tayo sa next. Um, sa microsporum naman, microsporum na mga organisms, well, wala na siyang space. Sige, tuloy natin dito. So, sa microsporum naman na mga individuals, meron siyang canis, we also have gypsum, and oduini. Hindi siya nabanggit dito, pero sige, lagay ko na lang din. Yan. Okay, so paano natin di-differentiate tong oduini from canis and gypsum? Okay. So yung sa M. canis, sa fluorescence natin siya i-check. Yung ectotrix hairs na sinasabi nila. So, differentiate M. canis from M. oduini. So, ito, canis from oduini. So, fluorescence, under sa fluorescence microscopy, si M. canis daw, green to yellow fluorescence. Si M. oduini naman, apple green fluorescence. May encounter nyo ulit tong apple green fluorescence sa IS. I think madi-discuss ko to soon. So, pag sinabing apple green fluorescence sa IS, it's primarily referring to fluorescence immunoassay. Anong degree of anong description daw ng color yung 2 plus ng fluorescence immunoassay. So, ang correct answer dun is apple green. Apple green fluorescence. Mayroon ding bright apple green and bright green fluorescence and brilliant green fluorescence. Pero sa kanya yun pag IS na. So yun. We also have here subcutaneous mycoses. So for subcutaneous mycoses, traumatic inoculation to skin or subcutaneous nga. So yan yung mga different diseases involving sa subcutaneous mycosis. We have here sporotrichosis or rose gardener's disease. The causative agent is Sporotrix shenkai. So dimorphic siya. Tandaan yan. Dimorphic si Sporotrix shenkai. So usually sa thorny plants. Yan. Yan rose. Wait lang. Wala. Sa Sporotrix shenkai, ang description na dito, according to my notes, sa mold form, floweret conidia. So, nagkakos siya ng Hello? Narinig ba ako? Nag-choppy ba ako kanina?
Sir, hindi po.
No po.
Hindi naman. Sige, sige. Okay, so floweret conidia. Sorry sa spelling. Ay, sorry sa sulat. So, yan yung mold phase niya under microscopy. Ang itsura daw niya is floweret conidia. Tapos, sa yeast phase naman, so recall natin anong dimorphic fungi. Di ba, it exhibits mold phase at 25 degrees and yeast phase at 37 in tissues. So, pag nasa loob siya ng tissues and pag 37 degrees, yeast form siya. Pag diphasic naman, mold at 25 and 37 degrees Celsius and yeast naman siya pag tissues. So, yun yung difference. Huwag niyong kalimutan. Yeast phase, cigar-shaped yeast cell. So, yan. Cigar-shaped yeast cell. Okay. Next, we also have Chromoblastomycosis. So, sa Chromoblastomycosis naman, demaciaceous fungi with the description of sclerotic bodies. So, wala na naman yung... Hello?
Hello po.
Wait lang. Idolo ko na naman yata yung PLDT. Saglit lang ha. Lipat lang ako ng data. For a while. Actually, pati data ko mahina din. Anyway, sige. Sana ma-push through pa rin to. So, pero naririnig niyo ba ako?
Yes, sir.
Okay, okay. Sige, sige. So, Chromoblastomycosis, yun. We have here Fonsesia species. We also have here Phyalophora and Cladosporium or Cladophyalophora. So, yung mga nakakainis na mga names niyan. So, yun. Ito yung mga differences ng Fonsesia, Phyalophora, and Cladosporium. So, ito, Cladosporium to eh. Itong Rhino, Flagella, wala naman siyang nabanggit dito sa side notes ko. Pero for Fonsesia, Pedrosoy, and Compacta, ang conidia nila, they are usually based on 3 categories. Wait lang. Hindi. Sa 3 categories. Yung Fonsesia dito, ang conidia nila is they are inside. At the side na lang. Kasi parang pangit pakinggan pag inside nasa loob nila. At the sides. And ang tawag dito sa phenomenon na to is at acrotica. So, anyway, I don't think this is a lot important, pero pangdagdag lang sa knowledge and side notes. Pag sinabi namang Phyalophora, wait. Okay, gets ko na. There are 3 classifications sa Fonsesia, na conidia nila. Medyo nakakalito nga lang kasi yun nga, ako nga rin mismo nalito lang ngayon. For conidia ng Fonsesia, we have at acrotican form. We also have, or acrotica form. We also have Phyalophora. So, yan. Iba yung Phyalophora species na Phyalophora na type of conidia. So, yun nga, nakakalito. Kala ko parang lang siya ng review center namin. So, Phyalophora. And we also have Cladosporium din. Cladosporium form, Phyalophora form, or type. Kaya bala. So, Acrotica form, Phyalophora form, Cladosporium form. For Acrotica, ang meron sa kanila, conidia nila at the sides. Phyllophora naman, Phyllophora form conidia in clusters. And Cladosporium naman, conidia in chains. So, yan. Pag sinabing Fonsesea, mixed sporulation siya. So, yun daw. Pag sinabing Fonsesea species, mixed sporulation. Pag sinabi namang Phyllophora, ito na yung species hindi yung form. Pag sinabing Phyllophora, only Phyllophora sporulation. Okay, gets. Sige, mali lang ng orientation yung review center namin. Ito yung types of conidia under sa Chromoblastomyces or Chromoblastomycosis. So, we have here Acrotica form, Phyllophora form, and Cladosporium form. So, Phyllophora na species, it only exhibits Phyllophora sporulation. So, ito, in clusters. Yung Cladosporium naman, exhibits, of course, Cladosporium sporulation. So, yun yung difference. Fonsesea, it can either be Acrotica, Phyllophora, Cladosporium, mixture of those. So, mixed sporulation daw. Phyllophora naman na species, they only exhibit Phyllophora, ito, sporulation. And Cladosporium only exhibits Cladosporium sporulation. So, for their drawing, ganito yung itsura nila. Sa Cladosporium type, ang itsura niya is something like this. Tapos, parang puno. Magba-branch out, ganyan. Ganyan yung sa Cladosporium. For for Acrotica, ganito. Sorry kung saan na nagda-drawing. So, for Acrotica, ganyan yung tsura niya. Acrotica yan. And for Phyllophora, parang bulaklak na rafflesia. Yan. So, ganyan yung tsura ng Phyllophora. So, Phyllophora ito. So, flower, yung itsura. Acrotica naman to. Form at the sides daw. So, ito. Ito yung hyphae, tapos at the sides nabubuo. Iba siya doon sa sessile ng Chlamydoconidia. Iba yun. We also have here Phyllophora in clusters. So, ito. In clusters siya dito. And sporulation, Cladosporium sporulation in chains. So, yan. Ganyan yung tsura niya. So, gets ba dito part or magulo pa rin? Sorry, ang gulo ng explanation ko kanina nung una. Gets na ba dito part? Hello?
Yes po, sir.
Okay. Sige. Anyway, kung kalimutan niyo na lahat ng sinabi ko dito, wag lang tong sclerotic bodies. So, copper-colored septate cells that appear to be dividing by binary fission. So, which of the following can be observed when the patient is suffering from Chromoblastomycosis? Ang una nating isipin na sagot, sclerotic bodies. So, for phaeohyphomycosis naman, ito, mas nakakainis din ng mga terms. Characterized by the presence of darkly pigmented septate hyphae in tissue. So, subcutaneous phaeohyphomycosis, Exophiala ginselmae, Bipolaris piscifera, Phyllophora richardshae, Wangiella dermatitidis. Then, cerebral phaeohyphomycosis naman, Cladophyalophora banciana. Anyway, for compre siguro medyo need na siyang basahin. But, when it comes to board exam, I don't think this is important. So, siguro for compre. Sabihin nyo na lang dito sa phaeohyphomycosis. Anyway, if may tatandaan man kayo dito sa slide na to, itong Banciana, Cladophyalophora banciana, causing cerebral phaeohyphomycosis. So, kumbaga, ang pwede niyang itanong sa compre niyan is, all of the following are considered as infections involving subcutaneous phaeohyphomycosis except for which species. So, yun. Pag lumabas to, ito yung sagot. Cladophyalophora banciana. Okay. So, yun lang din yung tandaan dito. So, mycetoma, yan. Chicharon. Eumycetoma or also known as maduromycosis or madura foot. So, mycetoma. Meron tayong tinatawag na actinomycetoma eh. Meron din tayong tinatawag na eumycetoma. Pag sinabing eumycetoma, true fungi causing disease. Pero pag sinabing actinomycetoma, tawag mo doon? Pag sinabing actinomycetoma, fungi-like disease. So, yun yung difference nila. Kasi nga, pag sinabing actinomycetes sila, nocardia yan, mga fungi-like bacteria. So, we also have here yung mga etiologic agents. Kumbaga, para may kinalaman siya sa phaeohyphomycosis, si Exophiala ginselmae. Yan. Ito. Ito si Pseudolesceria boydii, huwag niyong kakalimutan yan. Mycetoma. Madurella, basta may mga madurella, madura foot. Yun na yan. Then, Acremonium falciforme. So, yun. So, it may take greater than or equal to 4 weeks daw. So, medyo labag na siya dun sa general na storage natin for fungi, which is 21 days or from Baylis or 30 days. Pasok siya sa 30 days pero hindi na siya pasok sa standards ni Baylis. So, greater than or equal to 4 weeks daw. Okay, so proceed na tayo sa systemic mycoses. Imagine nyo yun, isang hapon lang natin ang second sem nyo sa medtech. So, yun. Systemic mycoses. We have here thermally dimorphic fungus. So, lahat ng mga nandito, lahat ng mababanggit na fungi dito, they are dimorphic. Again, recall, ano ang dimorphic? Mold at 25 degrees, yeast at 37, and tissues. Pag diphasic naman, mold at 25 and 37, yeast at tissues. So, yun yung difference. Okay, so infections are initiated in the lungs kasi usually ang mode of transmission nila is usually inhalation. So, yun. Sino ba nga bang organ ang una nilang tatamaan kundi ang lungs. So, ito yung according kay Jawets. Yan. Pero di-discuss natin. Okay, wala siya. Okay. Sige. Mag-side notes na lang ako dito. So, systemic mycoses. Dito na sa table. Though hindi siya complete. Pero sige. Blastomycosis is also known dito. Focus tayo dito. Also known as North American blastomycosis. North American blastomycosis or Gilchrist's disease. Caused by Blastomyces dermatitidis. So, blastomycosis or North American blastomycosis or Gilchrist's disease. So, ito. Blastomyces dermatitidis, mag-advance tayo ng konti. Pang boards. Ito, pang boards, sikretong malupit. Blastomyces dermatitidis, they resemble myelin globules in sputum. So, CM correlate. Bakit CM? Kasi ang sputum, microscopic examination ng sputum is under sa CM. Sputum. Yan. So, blastomyces dermatitidis. Ito ba nidlandan dyan? Yung mga places din nila actually medyo mahalaga din. Mississippi, Ohio, River Valleys. Yun nga yung weird eh. Kasi Southeastern, tama, North American. Meron tayong North America, South America. Tama. US is under North America, so yeah, tama. North American blastomycosis. Thick-walled yeast with broad base, usually single buds. So, hindi na mahalaga yan. So, proceed tayo sa next. We have also South American blastomycosis or yung itong paracoccidioidomycosis. So, South American blastomycosis. Yan yung other term niya. Isang tandaan nyo for Paracoccidioides brasiliensis. Isa lang need yung tandaan dito. Mariner's wheel appearance. Sa yeast phase yan yeast. Mariner's wheel. So, yan. So, for Paracoccidioides brasiliensis, mariner's wheel appearance. Then, we also have yung histoplasmosis or yung tinatawag nating medyo malandi yung pangalan disease, Darling's disease. Yan. So, Darling's disease. Histoplasma capsulatum. So, medyo need lang natin ng onting space. Gamitin na natin to for histoplasma capsulatum. Dito ko susulat yung mga niya, yung side notes. For histoplasma capsulatum, it resembles leishmania. Remember sa para? So it resembles leishmania. Nag-lecture na ba si Dinatan nakatoka sa parasite sa inyo eh? Nag-lecture na ba siya sa inyo?
Hindi pa po.
Ay, hindi pa? Oo. Ba't parang Anyway. So, yan. So, it resembles leishmania. The only difference is that sa histoplasma capsulatum, tawag mo dun? Sa histoplasma capsulatum, ang difference niya sa leishmania is that leishmania does not stain with fungal stain. Yan. Histoplasma capsulatum. It also resembles Cepedonium. Do you still remember Cepedonium kanina? Monomorphic fungi. So, yun. It also resembles Cepedonium. So, tuberculate macroconidia but no microconidia. Okay. For histoplasma capsulatum kasi meron siyang microconidia. Sa Cepedonium wala. So, yun yung difference. Pero, yun nga, pareho silang tuberculate macroconidia. Si histoplasma capsulatum also has tuberculate. Hirap nito pag pag puro sulat gamit mouse. Tuberculate macroconidia. Pero ang microconidia ni histoplasma capsulatum is pear-shaped. Pear-shaped microconidia. Then we also have for Cepedonium naman, tuberculate macroconidia but does not have microconidia. So, wag kayong malilito doon sa epidermophyton don't worry, mas madalas naman tinatanong sa boards ang epidermophyton. Wala din siyang microconidia. Ang difference niya kay Cepedonium is si microconidia club shape yung macroconidia niya. Si naman Cepedonium tuberculate. So, yun lang yung difference. And tawag mo doon, usually on cutaneous mycosis uh, epidermophyton. So, yun. Yan yung yan yung mga uh, another thing pa pala kay histoplasma. It exhibits positive results sa exoantigen test. So, which of the following fungi is positive sa exoantigen? Huwag na mag-isip ng iba, si Histoplasma capsulatum. H&M bands. Bands. O yan. Yan yung dine-demonstrate niya. H&M bands daw. Exoantigen tests. So, sige, kopyahin niyo muna saglit yung side notes. Okay na? Hello? Okay na ba? Proceed tayo sa next.
Okay po, kuya.
Opo, sir.
Sige. Okay. So, coccidioidomycosis naman, ang other term niya is San Joaquin Valley. San Joaquin Valley fever. So, coccidioides immitis. Ito yung need natin tandaan. Posadasi, medyo, hindi naman siya madalas tinatanong sa boards, pero si immitis, tandaan natin. So, yan. Another thing to consider sa coccidioidomycosis, tinanong siya sa boards. It is considered as major biologic hazard to lab personnel. Si Coccidioides immitis. Okay. So, yan. For Paracoccidioides brasiliensis, mariner's wheel appearance, yan yung need nyo tandaan. For Blastomyces dermatitidis, resembles myelin globules in sputum. Sa Coccidioides immitis naman, biologic hazard to lab personnel. And ang positive sa exoantigen test is Histoplasma capsulatum. Yan. Proceed tayo sa opportunistic mycoses. So, patient with compromised host defenses, doon lang sila nag-cause ng disease. Opportunistic ka, parang hinahanap na yung opportunity para mag-cause ng disease. So, yun yung ibig sabihin ng mga opportunistic. Mga mapagsamantala. Char. So, yun. Tawag mo doon, hinihintay nilang humina yung immune system bago sila mag-cause ng disease. So, yung mga diseases involved dito, sila candidiasis, cryptococcosis, aspergillosis, mucormycosis, and pneumocystis pneumonia. So, for candidiasis, premier cause of yeast infection in the world. So, ito daw yung ito yung need natin tandaan kay Candida albicans. Wait lang. And another thing din pala, actually marami yung side notes kay Candida albicans. For Candida albicans, it is considered as the only Candida that exhibits true hyphae. Yun. Only Candida that exhibits true hyphae. So, ayan. Primary fungal pathogen siya sa mga immunosuppressed patients, yung Candida albicans. So, positive siya sa germ tube. Na-discuss na natin yung sa germ tube kanina. Para sila ni Dubliniensis positive sa germ tube. Pero to differentiate further, dun na papasok yung morphology on cornmeal agar. So, yun. Si Candida glabrata naman, second most common Candida species. Ito yung saka mo sila mali-differentiate sa carbohydrate utilization test, yung mga Candida, different Candida species natin. So, yan, rapid assimilation of trehalose and glucose. Yan. Rapid assimilation of trehalose and glucose. And second most common Candida species to cause disease and account for 21% of all urinary yeast isolates. So first common, syempre si albicans. Yun. Rapid assimilation of trehalose and glucose. Yan yung sa carbohydrate utilization test. Candida tropicalis, not that much important. Parapsilosis, ganun din. And Crusade. So, ang tandaan nyo lang yung kay albicans at glabrata. Pati yung kay Dubliniensis with regards to germ tube. So, glabrata is negative sa germ tube. So, proceed na tayo sa cryptococcosis. We have here two etiologic agents, pero ang pinakatandaan natin si neoformans. Cryptococcus neoformans. So, possess large polysaccharide capsules. May mnemonics ako sa sa microorganisms exhibiting capsules which are infectious. So, ang mnemonics ko dun, some nasty killers have some capsular protection. So, ito. Some nasty killers have some capsular protection. Sino-sino sila? Salmon, tama. Wait lang. Streptococcus pneumoniae, nasty Neisseria meningitidis. So, Neisseria meningitidis. Killers, Klebsiella pneumoniae. Have Haemophilus influenzae. Some. So, ito na yung Salmonella. Or S. typhi. We also have capsular. Ito yung Cryptococcus. Protection, Proteus. So, ayan. Some nasty killers have some capsular. Hindi ko sinasabing sila lang yung merong capsule. Ang point ko is sila yung mga most pathogenic organisms that contain capsule. So, yan mga yan. Ano pa ba yung need nating tandaan dito sa slide? Wala, yan lang. Positive siya sa positive siya sa India Inc. Na-isolate siya sa CSF. Yan yung mga nitandaan kay Cryptococcus. Siyempre, yung kanina ay nabanggit ko, birdseed agar. So, yun. Birdseed agar. Ang creepy nung ibon kasi tumae ng seed. So, yun. Another thing to test sa CSF with the presence of Cryptococcus neoformans is yung cryptococcal antigen. So, yun. From the term itself, alam na natin kung para saan yung test na yan. Okay, so we also have aspergillosis. So, A fumigatus is the most common. Basta ang need natin tandaan sa aspergillosis is nagse-secrete sila ng aflatoxin. So yan. Produces abundant small conidia that are easily aerosolized and yun. So inhalation of fungal conidia, yan yung niya. Mode of transmission. Actually, yan lang need tandaan. Aflatoxin, A fumigatus, yun lang. Pati yung mode of transmission na rin. Pero dito meron din siyang lab test. Actually for me hindi siya mahalagang basahin. So skip niyo tong slide. So for mucormycosis naman, aka also known as Zygomycetes, is caused by molds classified in the order Mucorales. So yan. Kung maalala nyo kanina yung different different forms nila with regards to yung sporangiospores. So we have Rhizopus, we also have Absidia and Mucor. So yun. So ito yung mga different species involving yung Zygomycosis. Lichtimia, Kaning Hamela, Rhizopus. Yung mga yan. And ang need nating tandaan dito sa slide na to is Pneumocystis jirovecii. So, a typical fungus. Actually, ito yung major cause of death sa AIDS. Ito. Ay, shucks. Yan lang. Yan talaga yung number 1 na need natin tandaan kay Pneumocystis jirovecii. And pati na rin si Pneumocystis carinii. So, rodent form naman siya ng Pneumocystis. Okay, so before tayo proceed sa Viro, may questions ba tayo? May questions ba dyan? Hello?
Wala po, sir.
Wala naman? Okay, sige. Given kasi na ang myco, direct concept siya madalas. Actually, yung Viro, may at least somehow may konting analysis siya. Kaya hindi ko siya maituro na mabuti yung myco. Actually, confession lang to make. Ito yung isa sa pinakamahinang subject ko sa medtech. Mycoviro. Pero anyway, pumapalag naman kaya yan. Goods. So, yun lang. Yung mga sinulat ko kanina, siguro isi-send ko na sa inyo with annotations yung PDF ng PowerPoint para at least makopya nyo yung side notes. Pero isi-send ko na lang sa isa sa inyo. Particularly kay Za na lang. So, siguro let's have 20 minutes break. Goods ba sa inyo? 20 minutes break?
Yes, sir.
Yes,
Okay.
sir. Yes,
Okay.
sir.
General properties of viruses. Okay, so let's continue na. So we have here DNA viruses. Pero before that, magbigay muna tayo ng konting side notes sa mga different general characteristics ng viruses. So viruses are small in size, and they are what we consider as filtrable agents. Okay, mag-set up ako ng bagong for a while. Okay. So for virus, they are small in size, particularly 0.01 to 0.3 micrometers. Kung tatandaan natin, ang size ng HEPA filter, ang size ng HEPA filter na nagfi-filter ng substances is usually greater than 0.3 micrometer. So therefore, viruses can enter dun sa HEPA filter. That's why they are what we refer to as your filtrable agents. Yan. So filtrable agents because basically they can pass through filters. So yan, filtrable agents ang ating viruses. So why is this not much of a concern pagdating sa biosafety cabinet? So take note Ito for practical reasons to. There's no theoretical parang rationale. Pero for practical reasons, hindi masyadong concern na nakakapasok yung virus dun sa loob ng biosafety cabinet. Why? Take note na isa sa property ng viruses is that they cannot grow in a cellular media. So yung mga routine culture media na ginagamit natin for bacteria and fungi, actually hindi tumutubo doon ng viruses. They cannot grow on media without any cells involved. Kaya they cannot be parang it's also safe to say na hindi sila masyadong problem when it comes to contamination of growth ng mga bacteria kasi yun nga they cannot grow in an acellular culture media. So kahit makapasok sila dun sa loob ng biosafety cabinet, hindi sila magkakakos ng contamination sa culture media. So yan yung practical aspect ng bakit hindi masyadong concern yung viruses na nakakapasok sa biosafety cabinet. So yun. Viruses are filtrable agents nga ika nga nila. And yun. So there are some characteristics din na need nating tandaan diyan. We also have here yung either envelope ba sila or non-envelope. Non-envelope. So yung envelope, they contain lipid coating. Yung non-envelope, no lipid coating. No lipid coating. So yung envelope, they are heat sensitive or also ether sensitive. So, for non-envelope naman, they are ether resistant. Okay. So, there's also one thing that we need to know about them. Ito yung mga inclusion bodies, summarized inclusion bodies with regards to virology. So, meron tayong tinatawag na inclusion bodies such as negri bodies. So for negri bodies, usually pag sinabing negri bodies, isa lang isipin nating virus na nagkukos niyan, rabies. So rhabdovirus. So letter B, we also have here Torres Councilman bodies. Torres Councilman bodies. Torres dash Councilman bodies. This is caused by yellow fever. We also have here letter C, Guarnieri bodies. Guarnieri bodies. Caused by si pox virus. We also have here letter D, Bollinger bodies. Caused by foulpox. Then foulpox. We also have here letter E, cow-dry inclusion bodies. So ito medyo mahalagang tandaan to. So this is caused by herpes simplex virus or varicella zoster virus. So yan. Then next we also have here ito another important House eye inclusion. So sa CMV to. Yan. House eye inclusion. Then we also have here letter G, downy cell. EBV. Then we also have H. Another thing pala sa house eye CMV, HEMA correlate. So ang house eye naman sa HEMA, ang house eye appearance naman sa HEMA, it indicates Hodgkin's lymphoma. Kasi particularly, yan yung mapapansin natin sa sa Reed-Steinberg cells. Yan. Hodgkin's lymphoma. So Reed-Steinberg cells. And H, we also have here Koplik spots. So yan yung mga need natin tandaan. Koplik spots for rubiola or measles. Or morbillivirus. Yan. So, yan yung mga inclusion bodies na need nating tandaan with regards to virology. And yung mga specific na key viruses with regards to the most common, yung mga number 1 ganyan. So, mamaya sige. Negri bodies, rabies, Torres Councilman bodies, yellow fever, Guarnieri bodies, pox virus, Bollinger bodies, fowl pox, Cowdry inclusion, herpes simplex or varicella zoster. We also have house eye inclusion. We also have downy cells, Koplik spots. So, yung mga yan. And so yun nga, yung mga need nating tandaan na diseases involved for CMV. For CMV, number 1 congenital infection. So mag-involve ulit ako ng mnemonics dito. The microorganisms that cause congenital infection involves yung torches. Yan. So TO for toxoplasma, R for rubella, C for CMV. So actually siya yung siya yung most common na causative agent ng congenital infections. So number 1 siya, CMV. We also have herpes simplex virus, and yung S dyan, it's strongyloides. Yan. So yung mga yan yung nagko-cause ng or yan yung mga pwedeng mag-cause ng congenital infections. So, torches. Okay. Ano pa ba? Another is dun sa sa lab testing nila. So, nakopya niyo na ba lahat? Or may mga nagsusulat pa rin? Kopya na lahat?
Yes po, sir.
Okay, sige. So, yan yung mga need nating tandaan dyan sa mga yan. So, next, we have here yung different testing criteria with regards sa mga viruses. So, for storage, 4 degrees. We also have here for transport, we have here -70 degrees. for several days. Yan. Then we also have yung ano, pag serum for antibody testing naman, for antibody testing, negative 20 degrees. Yan. So, yan yung mga need nating tandaan sa ano, sa specimen criteria. And recall sa board exam namin. Recall ko sa board exam. Which of the following, so which of the following is most suitable specimen transport for viruses if we are going to transport them outside the lab? Hindi yan yung exact na tanong pero yan yung point. So, which of the following is most suitable specimen transport medium for viruses if we are going to transport them outside the lab? So, letter A, we have here agar plate. Letter B, we have here agar broth. Letter C, we have here yung ano, tawag mo dun? Sterile container. Letter D, container with container sealed with a sterile dry container. So sterile dry container and container sealed properly for transport. So yan. Actually mali ko to noon. Ang sinagot ko dito yung sterile dry container. Pero ang problema kasi parang nakaka-contribute din yung dryness ng container sa harsh environment ng virus. So pwede nila ikamatayan. So cross out na yan. Agar plate, all the more kasi hindi naman usually agar plate ang ginagamit pang transport. So agar broth naman, yung agar broth kasi is usually nilalagay yan sa tube. Tapos dyan sinususpend yung viruses. So correct answer is agar broth. Why? Take note yung mga different examples being used sa transport media ng viruses. Kung mapapansin nyo, ang isa sa transport media na ginagamit pa sa viruses would be Stewart's medium or Stewart-Amis medium. Hindi lang yan sa bacteria, pati rin yan sa viruses ginagamit din yan. And pag i-search nyo online kung anong itsura ng Stewart's medium, mukha siyang agar broth. So, the correct answer there is agar broth. Yan. Specimen considerations. Then, we also have here yung sa chick embryo. So, pag pinag-uusapan is chick embryo. Yan. So, pag sinabing chick embryo, they are used for rickettsia. So, ito yung mga different cell culture media na ginagamit for different viruses and other microorganisms. So chick embryo is for rickettsia, for tissue culture in short. So tissue culture. We have here A549 cells. These are used for these are derived from lung carcinoma. We also have here yung HeLa cells. So Henrietta Lacks cells. from cervical carcinoma. Then we also have yung Hep2 cells. Hep2 cells from laryngeal carcinoma. Hep2 cells, then human fibroblast cells. So human fibroblast cells, these are preferred for CMV. So wait, wag green. So they are preferred for CMV, yung human fibroblast cells. So A549 cells from lung carcinoma, HeLa cells cervical, Hep2 cells laryngeal, and human fibroblast CMV. So yan yung sa tissue culture natin. Okay, so actually yan lang yung need nating tandaan for general properties ng mga viruses. So proceed na tayo sa DNA viruses. Wait lang. Okay, so kita na ba?
Yes, sir.
Okay, so DNA viruses, tawag ba dun? Ito yung mga different DNA viruses, naked DNA viruses. So parvo, papilloma, polyoma. So inaswan na yung actually inaswan na yung tawag ba dun? Inaswan na yung papilloma and polyoma. Papova na yung tawag. So Papova viridae, adenoviridae, and hepab na viridae. Wait lang. So si papilloma and polyoma inaswan na, Papova na yung tawag, Papova viridae. So yun. Ito medyo lumang reference na itong gamit ni Sir Raymel dito. Pero anyway, for DNA viruses, ang mga classifications dyan is that all replicate in the nucleus except Pax viridae. Yan. So wait lang. I think may mga corrections ako dito for a while. Okay. Tama. So all replicate in the nucleus except Pax viridae. So for DNA viruses, ang mnemonics natin dito ay hepapaad po. Papaad po. Yan. So herpes virus, hepadnaviridae. So herpes viridae, hepadnaviridae, parvoviridae, papova viridae. So papova is papilloma and polyoma. Tapos we also have adenoviridae and Pax viridae. So yan. All of them replicate in the nucleus except Pax viridae. Largest DNA virus known to man is Pax viridae also. Tapos ano pa ba? Yun. Then lahat sila they are icosahedral except for Pax viridae din. Complex. So for Pax viridae, it is also complex. Yan. Ang smallest naman dito is yung parvoviridae. Smallest. Okay. So, yan. For dito sa PowerPoint slide naman, Parvoviridae is also the causative agent of Erythema infectiosum or fifth disease. Si Parvovirus B19. So, siya yung nagkakos niyan, yung Erythema infectiosum or fifth disease. So, kung babalikan natin lahat ng six disease, yung mga yan tinutukoy na fifth disease, sixth disease, yung mga yan, yan yung collectively known as childhood exanthems. So, childhood exanthems. So, for childhood exanthems, ang classical characteristics ng mga diseases under dyan sa childhood exanthems na yan is yung tinatawag nating mga rashes. So, bukod dun sa rashes, after rashes, parang redness ng skin. So, erythema. Yun din yung classical feature ng ng childhood exanthems. So, the first disease is yung rubella. Rubella. Second is yung scarlet fever. Next is yung rubeola or common measles. Next, we have here yung hindi, baliktad. Rubeola yung first. Rubella yung third. Then we have here yung ikaapat, staphylococcal scalded skin syndrome. So, staphylococcal scalded skin syndrome or Reiter's syndrome. Then yung fifth, yan na, si Erythema infectiosum. And six is Rosola infantum. Rosola infantum. So, 6 disease yan. Caused by human herpes virus 16. HHV6. So, scarlet fever caused by Streptococcus pyogenes, rubeola or yan. Tapos rubella, then Staphylococcus scalded skin syndrome, Erythema infectiosum, tapos Rosola infantum. Ayan. Papilloma viridae, human papilloma virus or warts in humans. Polyoma naman is yung basta tumor siya caused by JCV. So John Cunningham virus and BKV. John Cunningham virus. Yan, tama. Adenoviridae naman is infection of the respiratory tract. So for adenoviridae, isa din yan sa may side notes ako. Number 1 siya sa viral conjunctivitis. Ito. So adenovirus, number 1 sa conjunctivitis. Yan. We also have yung Hepadnaviridae. So, ito si Hepa B. Or serum hepatitis and chronic. Ang infectious hepatitis yan, si Hepa C. So, yan. HB-Sag is present in early disease. Anti-HB-Sag indicates immunity. Di ko alam kung matatakal natin to sa undersakin din sa serology soon. So, pero yun nga. I-discuss ko siya pag may time. Itong different markers, yung table niya sa Hepa B. Kung kailan tumataas si ganito, kung kailan bumababa si ganito. So, yan. We have also here envelope DNA virus with icosahedral symmetry, herpes viridae. So, hindi ko nabanggit kanina yung different envelope viruses sa DNA. So, pag envelope, meron dyan si Hehe po. So, Hepadna, herpes, and pox virus. Pag non-envelope naman, or naked, meron dyan si paadpa. So, parvovirus, parvoviridae, adenoviridae, and papovaviridae. So, yung mga yan. Hehe po, envelope, paadpa, non-envelope. So, yan. Ito sila herpes virus to. So, HSV1, positive siya sa chunk cell test. So, yung HSV1, ang palatandaan dyan kung saan siya nag-infect, kung sa oral cavity ba or sa genital tract, magbe-base kayo doon sa number. So, syempre, si number 1, siya yung pinakauna na sa taas. So, oral. HSV1 is oral. HSV2 is genital. So, yan. Pero HSV2 is also reported in some cases sa oral. So, it's also safe to say na si HSV2 can be found in the genital tract and oral cavity. Pero si HSV1 sa oral cavity siya. So, yun yung need natin consider kay HSV 1 and 2. Tapos, for HSV1, siya din yung causative agent ng cold sores. This is also known as your herpes labialis. Herpes labialis. Okay. So, HSV2, HSV3, ito. HHV3, sorry. Human herpes virus number 3 to. So, varicella zoster. Ito, HHV1, HHV2, then HHV3, varicella zoster virus, chickenpox. So, mild self-limiting lang siya, usually sa children. Tapos pag pag naging matanda ka na nagkaroon ka ng chickenpox dati nung bata ka, pag tumanda ka na at humina ang resistensya mo, magre-reactivate yung mga yan to form your varicella zoster virus, which causes shingles. So, yan. For varicella zoster virus, tawag mo dun, reference method na ginagamit dyan is your fluorescent antibody membrane antigen. Membrane antigen. So, FAMA. Yan. So, fluorescent antibody membrane antigen sa varicella zoster virus. Pero ang most common and easiest method, most common and easiest is ELISA. Yan yung kay varicella zoster virus. Okay. Next, we have also beta herpes viridae. Ang mahalaga dito is ito. Pero si CMV under siya sa human herpes virus number 5. Ito formerly known as salivary gland virus. Or ito din, it causes kasi 40-day fever. 40-day fever. Yan. So salivary gland virus or 40-day fever, si cytomegalovirus. So, ano yung alam natin kay cytomegalovirus or CMV? Siya yung number 1 na most common na congenital infection worldwide. Siya din yung nagko-cause ng owl's eye inclusion sa inclusion body sa sa cells natin, CCMV. So yan. Human herpes virus number 5 siya. Then we also have yung Epstein-Barr virus. Huwag niyo napansin yung mga beta, gamma, ganyan. Hindi naman mahalaga yan. Yung Epstein-Barr virus naman, human herpes virus 4. This one is the one that causes kissing disease. So yan. Atypical lymphocytes and heterophile antibodies. Check natin kung mahabol natin na ma-discuss to soon. And it is also associated with Burkitt's lymphoma. So Burkitt's lymphoma is yung sa hima, kung maalala natin, acute lymphoblastic leukemia 3. So or L3 in short. Yan yung Burkitt's lymphoma. So acute lymphoblastic leukemia 3. Type 3. Human herpes virus 6, yan, exanthem subitum or Rosola infantum or 6 disease. Exanthem subitum. So, yung nabanggit natin kanina, childhood exanthem, siya yung ika-6. We also have here Pax viridae. So, naked siya. Diba? Non-enveloped virus, hehepo. So, wait. Naked ba siya? For a while, for a while. Tama, tama. For a while lang, for a while. Kasi yung alam ko, siya envelope. Wait lang. Okay. So, sakto. Siya? Envelope siya. Disregard this one. Actually, siya? Envelope. Confirmed by Jowetts. Yan. So, stick pa rin tayo dun sa, sa mnemonics na hehepo. So, yun. Envelope viruses siya. So, ang need natin tandaan for Pax viridae is yung cowpox or tinatawag nating vaccine niya. Then, yung variola. So, we have here variola major and variola minor. Variola major nagkukos ng smallpox. Variola minor is nagkukos ng tinatawag nating alastrim. So, yan. Yan lang need natin tandaan for variola. Molluscum contagiosum naman, small wart-like lesions. So, usually they mimic genital herpes. Okay. So, proceed na tayo sa RNA. Are there any questions regarding sa sa DNA viruses? Sorry sa confusion kanina, pero stick pa rin tayo dun sa rule na na hehepo. May questions ba regarding sa DNA? So, malapit na tayo matapos. RNA viruses na lang. 10 slides pa. Hello?
Hello, sir.
May questions ba regarding sa DNA viruses?
Wala po, sir.
Okay. Sige. Proceed na tayo sa RNA. So, ito na yung last portion ng ng Mycoviro topic natin. So, envelope RNA viruses with sige, before mo tayo mag-stick diyan, proceed muna tayo sa characteristics, general characteristics nila. So, yun. They are generally, hindi ko sinasabing lahat. So, generally, ssRNA, single-stranded, helical, envelope, and they replicate within cytoplasm. So, yan yung general characteristics nila. Siyempre, may mga exemption yan. So, for dsRNA, yung double-stranded RNA, we have the following. Double-stranded RNA involves Rheoviridae. So, we also have yung icosahedral. Picorna, so PCR, RTR yung mnemonics ko dyan. Pero pangit na mnemonics yun. We have here Picorna viridae. Calisi viridae. So, Calisi viridae. We also have Rheoviridae. We also have Retroviridae. So, dito sila HIV. We also have yung Toga viridae. And Flaviviridae. So, yan. RTF pala, sorry. So, Picorna viridae, Calisi viridae, Rheoviridae, Retroviridae, Toga viridae, Flaviviridae. Yan yung mga icosahedral. We also have yung naked or non-envelope. Di ba kanina they are generally ssRNA, single-stranded, helical, envelope, replicate within cytoplasm. So, ito yung mga exemptions. dsRNA si Rheoviridae, then non-envelope or naked si PCR, Picorna viridae. Actually, yung first three na nabanggit dito sa icosahedral. So, Picorna, Calisi, and Rheoviridae. So, Picorna, Calisi viridae, and Rheoviridae. Then, these are the ones that replicate within the nucleus. Kanina, general natin is cytoplasm. Ngayon naman, nucleus, orthomixo. And we also have yung retroviridae. So, yan. Yan yung mga general characteristics nila. Smallest RNA virus is found in Picorna kasi nga from the term pico, yung unit of measurement na prefix, pico, maliit, RNA. So, Picorna viridae. And yung smallest virus dyan is si enterovirus. Sa Picorna viridae siya. So, yan. Largest naman si Paramyxo. Okay. So, now let's proceed muna sa orthomyxoviruses. Ano yung palatandaan nito sa kanya? It replicates in the nucleus. So, yun. It replicates in the nucleus and i-copy paste na natin yung ibang ano niya, yung ibang general characteristics niya. So, yun. Yun yung kay orthomyxoviridae. So, influenza viruses, we have types A, B, and C. Ang most common is type A. Ang least common is type C. Least common type of influenza. So, most common si type A. We also have here race syndrome associated with influenza type B. Tapos, diagnosis involves hemagglutination inhibition test. So, more details later on sa sa IS natin. Pero when we say hemagglutination inhibition test, kumbaga yung soluble antigen is allowed to react with tawag mo doon? Soluble antigen is allowed to react with particulate antibody. So, yun. And then, pagka nag-react siya sa particulate antibody, next na ilalagay dyan is yung RBC containing the containing the antigen. Wait. Tama. Containing the antigen. Ngayon, pag the antigen will, pag the soluble antigen will bind to the particulate antibody, pag nag-bind sila together, wala nang extra na space na magbabind sa RBC. So therefore, ang mangyayari dyan is walang agglutination. Kapag kapag walang soluble antigen involved, yung particulate antibody, magbabind siya dun sa RBC. So therefore, may agglutination na may involved. So yun yung sa hemagglutination inhibition test. Ulitin ko lang ha. Wait. So dito. Let's say ito yung soluble antigen found sa patient. Yan. Then ito yung particulate antibody. Yan. So tapos may third reagent which is yung indicator cell, yung red blood cell. So pag magbabind yan, kapag present yung antigen dun sa patient, magbabind siya dun sa antibody found sa particulate antibody or reagent antibody. And then pag nagbind siya diyan, hindi na pwede magbind tong particulate antibody dito sa red blood cell, causing inhibition of agglutination. And pag mayroong inhibition ng agglutination, positive result yun. So, yun. Pag wala naman tong soluble antigen, the binding sites of antibody found in this particle is able to bind dun sa antigen found sa RBC. Kaya pag nag-bind siya dyan, merong presence ng agglutination, hence negative result. So, yun yung hemagglutination inhibition reaction. I-discuss ko to later on sa IS. More details. So, yun. Let's proceed sa Paramyxoviridae. So, we have here para-influenza and mumps. So, para-influenza, it causes croup in infants. Yan. And mumps, generalized disease associated with enlargement of parotid glands or what we refer to as parotitis. So, there are some cases wherein yung mumps, it may cause, saglit lang. Yung mumps, it may cause orchitis in men. So, pag sinabing orchitis, or parang magiging infertile yung lalaki. So, it can cause infertility sa lalaki, yung mumps. Pero ang most common cause of infertility sa mga lalaki, so balik tayo sa CM. Most common cause, lumabas to sa March 2021 board exam. is yung varicocele. Yan. So, most common cause of infertility is yung varicocele. So, morbillivirus associated with measles virus can also cause Koplik spot. So, maliban sa measles virus, morbillivirus can also cause Koplik spot. Pero mas common si measles virus. Pneumovirus associated for bronchiolitis and pneumonia in infants. So, respiratory syncytial virus yan. So, yan. So, hindi na mahalaga yan. Arena viridae, ito yung lasa, ito yung with regards sa hemorrhagic fevers natin. Si Arena viridae. Tama, Lassa fever. We also have yung rhabdoviruses or bullet-shaped viruses. Yan na sila rabies. Yan, na-discuss naman na yan, negribodies. Tapos, we also have yung coronaviruses. So, ito yung given na nung 2018 pa yata ginawa ni Sir Raymel to. Pero, itong coronaviruses, under dito si SARS-CoV-2. Siyempre. Yan. Or COVID-19. Bunyaviridae or we also have your hantavirus, lacrosse virus, and California virus. So, ano yung mga side notes dito? Hantavirus. Wala masyado. So, yun. California and lacrosse viruses, wala masyado. As in, straight to the point lang talaga ang Mycoviro eh. Pero sige. We also have here yung yung tinatawag natin na arbovirus. So, arboviruses, arthropod-borne. So, arthropod. We have here Bunyaviridae. Except hantavirus. So hindi arthropod-borne si hantavirus. So hantavirus is not arthropod-borne. So yan. We also have yung Flaviviridae. So sila Ebola virus yan. Marburg virus, yan, mga yan. Then we also have Togaviridae. Hindi yan Jayati yan. Togaviridae. Okay. So, need natin tandaan kay Togaviridae, Eastern equine, Western equine, yung mga yan. So, yan, arbovirus, arthropod-borne. So, mosquito vectors and animal reservoirs. Rubivirus, rubella, causing German measles. So, yan. Basically, direct terms lang talaga. So, kaya nga hindi ko masyadong dinidiscuss mabuti itong Mycoviro kasi in the first place, 5 items siya madalas sa board exam. Combined na yun, Mycoviro. Kaya yung mga malakas na kutob na pwede lumabas sa board exam and compre yung mga nibibigyan ko ng highlight. So, yun. Flaviviridae, dengue virus, arbovirus transmitted by mosquitoes. So, yan mga yan. Hepatitis C. Well, actually, ano siya eh. Wait lang. Okay. Tama. So, hepatitis C, flaviviridae. Yan. Yellow fever, West Nile, ande silang lahat kay Flavi. Sorry. Yung kanina nabanggit ko na Flavi, si Ebola, ande pala siya sa philo. So, Filoviridae sila, Ebola. Yung Flavi sila, no, sila dengue virus, yellow fever, ganyan. So, from the term Flavi, meaning yellow, so under na din si yellow fever. And si Marburg, under siya sa Filoviridae. So, RNA virus, we also have Retroviridae. So, isa sa general characteristics nila, unique characteristic nila is they have reverse transcriptase enzyme. So, yung reverse transcriptase enzyme, from the term reverse transcriptase, kino-convert niya from RNA to DNA. Contrary dun sa normal na central dogma of molecular biology na from DNA to RNA to form yung transcription process. So, yun. So, full-blown AIDS, yan yung need nating tandaan. Kaposis sarcoma. So, pneumocystis jirovecii, so yan. Yung na-discuss natin kanina na most common cause of death sa AIDS patients, pneumocystis jirovecii. Ito tanong siya sa boards. Which of the following antibodies are first produced upon the presence of HIV infection? So, ang sagot dun is antibody to P24. Siya yung first na napoproduce na antibody. Yan. Then we also have human T-cell leukemia virus, so HTLV1. Wala masyadong ano dyan. Okay, so picorna viruses. Enterovirus, ang need nating tandaan dito is ito yung isa sa causative agent ng aseptic meningitis. Yan. Si enteroviruses. Yan, oh. So, pag mayroong viral meningitis, ang usually suspect dyan si enterovirus. Under si poliovirus sa enterovirus. Pero, ang main feature ni poliovirus is poliomyelitis. So, yan. So, we also have yung Coxsackie virus type A, hand, foot, mouth disease. Sige. Yung hepatitis A dito, nabanggit siya as infectious hepatitis. Pero, kino-consider din si Hep C as also infectious hepatitis. Through, not through fecal route nga lang, but also through, through parenteral route or blood. So, yun. So, rhinoviruses, most common cause of the common cold. Yan lang need natin tandaan dyan. Acute infantile diarrhea. Causative agent, si rotavirus. So, yun. Basically, yan na yung end ng topic natin. So, straight to the point lang talaga ang Viro and Myco. Any question?
Wala po, sir.
Okay. So, yun. If ever man na, na magbibigay ng priority sa microbiology, yun nga, tulad ng sinabi ko kanina, ang prioritize nyo is yung yung, tawag ba dun? Ang prioritize nyo is yung bacte, followed by para, then mycoviro. So, yun yung suggested na order ng priority when it comes to microbio. Pagdating sa board exam. Pero yan kasi sa comprehensive exam nyo, 1,500 items yun, which consists of 15 subjects, 100 items each. So separate yan. Bakte, separate ang bakte sa sa para, separate ang para sa Mycoviro in AS1 yan. Tapos we also have CC1, CC2, CC3. Separate yung mga yan. Tapos HEMA 1, HEMA 2, immunology, serology, tapos blood banking, then automation, MTLB, histopath, ano ba? Actually, separate ata ang immunology sa serology. So, yung mga yun, 15 subjects lahat yun, 1,500 items. And to pass the comprehensive exam, you need to get at least 750 items. Minsan, it depends na lang, discretion ng dean or sila Sir Jack kung magbibigay sila ng adjustments just in case na konti lang ang nakapasa. So, usually 60% nga dapat. So, 900 items dapat ang makuha nyo para makapasa kayo sa compre. So, malay ba natin kung yung knowledge nyo ngayon sa Mycoviro, yung 1 point dun nakasalba sa inyo sa comprehensive exam para maging 750, o diba? Saya sa feeling nun. Makakuha ka lang ng 750 points, sobrang saya na yun. At least 50%.